Okay. And then for this chapter 13, is actually the important point is you need to know about how you describe the excretions, and also for this requires the structure of the nephrons. You know the nephrons? So inside the nephron, right, they are comprised of the glomerulus, okay, and the proximal convolutubule, and then the distal convolutubule, and also loop Henle. So let's talk about this glomerulus first. So glomerulus, right, is actually just like a network of the capillary. Okay, it's something like this. Okay, it didn't show in this picture. Yeah, okay. See, A will be the glomerulus. Okay, so actually glomerulus is just like blood capillary. A lot of blood capillary, they join together, form a network. So this is called as a glomerulus. So what's important for the glomerulus? So this glomerulus, right, actually they are come from where? They are come from this X. X is referred to this one, we call it the afferent arteriole, okay? X. Afferent arteriole. So afferent arteriole is, the blood is come from where? The blood is come from the renal artery. Okay, the blood is come from this renal artery. So it will be the renal artery. Pass the blood. They are transport the blood to the afferent arteriole. So the afferent arteriole, they will transport the blood reaching the glomerulus. So between the glomerulus, right, and also the bone capsule, there will be required as a filtration membranes. Okay, so inside here will be the bone capsule. This space. Okay, bone capsule is they are referring to the space. Bone capsule. Because between glomerulus and this bone capsule, right, there will be a membrane. So this membrane, they have a very, very small pore. So they only allow the small particle able to diffuse into the bone capsules and then go to the proximal convolutubule. So this will be the proximal convolutubule. This part B. Okay, so this will be the process. So for the glomerulus, right, you need to know how to write the structure, okay? It's a capillary and it's ball shaped and it's very thin, okay? Just like the alveolus, just something like alveolus also, very one cell thick. So they are actually provide the blood at a very, very high pressure. Do you know why they're able to provide the blood at a very high pressure? It's because the afferent arteriole has a larger diameter, okay? Due to the larger diameter of the afferent arteriole. Oh, okay. Due to the larger diameter of the afferent arteriole. You know, right? Afferent arteriole, right? They have a very large diameter, so they can transport more blood enter into the glomerulus. So due to this feature, right? So the blood, they can be transported in a very high pressure, and plus this blood is originated from the renal artery. So artery, right? Artery, they always transport the blood in a much more higher pressures, and also they are very, they have a lot, we call it small loop, right? So they can help to provide a very large surface area. So they can allow the ultrafiltration to occur. Ultra means what? Ultra means that very fine, very small particle. Filtration means that you help to filter the particle. So the small molecule able to filter out of the glomerulus, right? And then they can enter into the bone capsule. So later they will go into the bone capsule. So only the large molecule, like they stay in the glomerulus, and then they will go back to the afferent arteriole. Okay, so from the this glomerulus, right, they will enter into the bone capsule. Okay, they will get into the bone capsule. So you should know what kind of particle can get in the bone capsule. Is referred to this one. Okay, the small particle side, which is this water, salt, glucose, amino acids. They will get into the bone capsule. So how about the large particle sites, like the plasma proteins, okay, our red blood cell, and also the platelets? Their size is too large, right? So they cannot pass through the membrane, enter into the bone capsule. They cannot. Yeah. So this is the importance of the process about the ultrafiltrations. occur at the glomerulus. Okay, so they will ask you what kind of molecule they can reabsorb in the kidney tubule. So it will be this glucose and amino acids. Okay, that's all. And then for the liver is the very important organs. Liver, they can help in do what? They can help in do the metabolism. Okay, they can help to do the. I mean, when we talk about function of liver, right? They can do the metabolism. The metabolism is they including the carbohydrates metabolisms, proteins, and also the lipids. Okay, for example, the metabolisms like the carbohydrate includes, they can help to convert the excess glucose and store in the glycogen inside the livers. So this is an example of the functions. So apart from that, liver, right, they can do metabolism, they can do a detoxification also. Detoxification. Okay, and also they can do what? They can do the, we call it assimilation. Assimilation, all right. And they can do the excretions. And also the deamination. Those are processes occur in the liver only. So what is the role of liver in the excretion? When you talk about excretion, right? Excretion is referred to the removal of the waste product, include those things, okay? Like the carbon the carbon dioxide, the urea, feces, mineral salt, that kind of thing. So when you talk about liver in the excretion, okay, so actually it's talk about the deamination. Okay, deaminations. Because one of the waste product being produced by your body is a urea. So how your body able to produce a urea? So it's from the process of the deaminations. So this deamination of amino acid, what they do is they actually remove the nitrogen group away, okay, from the amino acids. So you just imagine, right? Do you still remember about the structure of amino acids? Okay, amino acid, right, is kind of the structure like this. They have C, and then they have two functional group. One is we call as a carboxylic group. They have this. Another one we call as a NH2, amino group R and the H. So this will be the amino acids. Okay, this will be the amino acids. But what happens during the deamination is you actually remove this functional group away. We call it as the amine group. Okay, you actually remove this amine group. So removal of the nitrogen containing part of the amino acid, or sometimes we call it remove the amine group. So after that, this amino acid, right, after they being removed, they will become the ammonia. So from here, they will become the ammonia, okay? Ammonia, the structure is NH3. Okay, NH3. But the problem is, ammonia itself is actually toxic. Okay, ammonia is toxic substance to the body. So you need to convert this ammonia become something which is less toxic, which is a urea. So that's why ammonia, which is a toxic one, they will convert become a urea inside the liver by the detoxification. Okay, ammonia toxic, they will convert to urea, which is the less toxic one. So that's why liver can help to break down any toxin drugs when they receive the blood from the outside. Okay, so they help in the deaminations to produce ureas. So to produce urea from the ammonia. Ammonia is from the amine group, okay, from the removal of the nitrogen containing groups from the amino acid. So liver, okay, apart from that, they can help to do the detoxifications, which is they can help to break down the toxin and the drugs. And also they can break down the worn out red blood cell. Okay, because the red blood cell is being damaged, right? Okay, what's meant by worn out red blood cell? Worn out red blood cell is referred to the red blood cell, maybe it's being damaged by some others, for example, some other virus or pathogens. And also some high red blood cell, they are aging. you know, the aging red blood cell, because red blood cell is a cell which they do not contain nucleus, right? So, and also red blood cell are actually, they have very short lifespan only. They only survive in our body about 120 days. Okay, so red blood cell, they were aging, right? So slowly they will worn out. So that's why this worn out red blood cell, they need to be, they need to be removed away from the body, because they are not become a functional cell anymore. That's why, so how can they be removed? So they need to be transferred back to the liver and then to the spleens, so they can help to destroy the worn out red blood cell. And then the liver produces bile. Okay, so that's it. And also, you need to know about the structure. Okay, label the structure. You can see this. So they will show you an arrow. Okay, this one arrow. So this arrow, right, when the blood coming, it will be referred to the renal artery. Okay, so the renal artery, they will enter to the afferent arteriole and then get into the nephrons. Yeah. And then this K, okay, the kidney, right, they have a outer covering surface, we call it as a renal cortex. Okay, covering surface like that. So the inner part will be the this one, renal medulla. Okay, so inside this renal medulla, right, they can find a lot of the functional nephrons. A lot of them are situated on these regions. And then inside will be the renal pelvis, which is at this area. Here will be the renal pelvis. Okay, and then and it will refer to the, we call as a ureter. This ureter, right, they can help to transport these urines from the kidney, enter into the bladder. Okay, so what kind of waste product that can be removed away from the blood by the dialysis? Do you know what the dialysis? The dialysis is kind of machine. Okay, so today we talk about the dialysis. Okay, let me show you about this dialysis. We almost finished chapter 13. Okay, dialysis machine. Okay, so at the maybe you can imagine, okay, if one day you have a kidney failure, okay, means that your kidney cannot work properly. So what will happen to you? So means that your body unable to remove the toxic substance within in your blood. They cannot be removed away. That's why you need the assistance from the machine, okay, to help you to move away the waste products from your blood. So if both of your kidney being damaged, right, usually there will be no treatments, because, and if your kidney being damaged, right, so you'll be like slowly, you'll be slowly die off. Like, it's because that the toxins and the urea and a lot of waste products is removed. It's being accumulated inside your body. So they will build up inside your body. So that's why it will cause something like that there's a condition. Okay, there's conditions due to the accumulation of toxins inside your body. Okay, so that's why you need the help from the kidney dialysis. Kidney dialysis actually is an artificial kidney machines help to carry out the functions of the kidney outside the body. So actually this machine, right, they help to replace the kidney, okay, or sometimes you can do the kidney transplants. Means that, but you need someone to donate the kidney for you, okay. And usually this kidney transplant, right, is not 100% successful. It's because that if your organs, the blood type is not matched, okay, sometimes they will have this kind of, like, we call it as the reactions. Okay, reactions will occur. So usually they will do this kidney dialysis. Okay, they will get the help from the kidney dialysis for the kidney failure patients. Kidney dialysis, so it's an artificial method help to filtering the blood to remove the toxins and the excess substance away. So usually, right, when you want to use this kidney dialysis, right, usually they help to use we call catheters to connect the tubes and enters into your artery for your body. So actually, you are connected to the dialysis machines. So this dialysis machine, right, it just functions just exactly same to the kidney. So what they do is they will help to remove most of the ureas and the waste products inside the blood. and hence maintain the water and salt balance of the blood. Okay, this is very important. It's because that you need to maintain your, your osmotic pressures inside your blood. Okay, so how they work is this unfiltered blood, right, is actually taken out from the artery in the arms, and then they'll pump the blood into the dialysis machines, and then return to the veins in the arms. So inside the machine, right, the blood and dialysis fluids are being separated by the partially permeable membranes. So the blood will flow in the opposite directions to the dialysis fluids, allowing the exchange occur between the concentrated gradient. So inside the fluids, okay, inside the fluids, they just acting like the body fluids. Okay, because body fluid, right, inside of body fluid, we also have the acids. We also have the water and some salt. Okay, so the dialysis fluid inside, they also contain the glucose, salts, and the glucose salt and some mineral salt. Yeah, but there will be no urea. Okay, because urea is considered as a waste product. So this is like in this way. Yeah, you can see. This is the machine, right? Okay, so actually the blood is being traveled from here. I mean the renal artery. The renal artery will be connected to these sides. So they will help to pump the blood enter into the dialysis machines and then pass through the machines and get out. So you can see, right, it's very connected. I mean very close connected to this fresh dialysis fluids. So what happened is osmosis occur, okay? Osmosis occur, which is involved in the movement of water, but the most important is simple diffusion occur, okay? So what happened is because that inside the blood, right? Inside the blood, since that you don't have your kidney help to remove the waste product from your blood, so inside your blood, there must be a very, very high level of the toxin accumulations, correct? So example of toxic accumulations inside your blood is maybe the urea, maybe either of the ions, for example, like the potassium ions. So here they must be very high concentrations here. They must be very high concentrations inside the blood as compared to the dialysis fluid. So that's why here become high concentration, right? Then the dialysis fluid become very low concentration. So it will cause the urea, okay, urea being transport from the blood, which is a very high concentrations, enter into the dialysis fluid, which have a very low concentrations. So here's a method, okay, which is by the simple diffusion. So after this dialysis fluid, right, they get the waste products from the blood cell, and then they will transport the waste products away, okay, together with the dialysis fluid. Okay, so what they do is they will transport the clean blood back to the body of the patients. Okay. So just they need to ensure the blood, all of the blood pass through the dialysis fluid, so they can get rid of most of the waste products enter into the dialysis fluids. Okay, so when the blood, they just pass through the dialysis machine, right? So when they, when the blood being released out, they'll become a clean blood, okay, clean blood without any the waste bacteria. So they will go back to the patients by the vein. So about this process, right, you also have this, we call it an anticoagulants. Okay, this anticoagulant is very, very important. It's because that your blood tend to have the, we call it coagulators. Okay, something like blood clot. Okay, so that's why you need to have some anticoagulation, because you want to prevent the blood clotting. Because if the blood is being clot, right, your blood flow will not be smooth. Okay, and then they are unable to enter into the dialysis machine because they are being clot. So as we know that dialysis fluid, right, inside they don't have the urea. They have very high concentration gradient. So that's why the urea just diffuse through the membrane from the blood enter to the fluids. As the fluid, right, they contain the glucose concentration, which is equal to the normal sugar level. Okay, so this will prevent the net movement of the glucose across the membrane. So you can see, right, they will just maintain the glucose concentrations inside the blood, just same with the dialysis fluid. So they can help to maintain the concentrate gradient because they want to prevent the glucose, right? The glucose also being diffused from the blood and enter to the dialysis fluid. Okay, so they want to prevent this happen because they just want to get the waste product only. Glucose is not considered as a waste product. Waste product is something like the urea and some salt and ions. So also the dialysis fluid, right, they also contain the salt concentrations just similar to the ideal blood concentration. So the movement of the salts across the membrane only occurs when there's an imbalance. Okay, means that if the blood is too low in salt, right, so they will diffuse into the blood. Means the salt from the dialysis fluids will get into the blood if the blood really lacking of the salt. Or sometimes if the blood is too high in salt, right, they will diffuse out of the blood, enter into the fluid machine. So that's why the fluid in the machine, right, is always continuously refreshed so that the concentrate gradients are maintained between the dialysis fluids and the bloods. Okay, so when you want to do this dialysis, right, okay, usually you take about 3 to 4 hours to complete. Okay, and then for the kidney failure patients, right, they need to always visit to hospital because they need to do this kidney dialysis. So usually this is something like it's not a treatment, but they can help to reduce, they help to control the problem. So usually being done several times in a week to prevent damage of the toxins to the body. Okay, because your body, right, is very sensitive toward the toxins. So you want to try your best to remove the toxin away. So if the toxin occur in your body, right, it will cause, it will cause a lot of side effect. Okay, so this is kidney dialysis. And how about kidney transplant? Okay, kidney transplant, right, you need to ensure the matching of the blood type. Also, you want to avoid because the immune response happens. Okay, leading to the risk of the rejections. Okay, so if the rejection occur during the organ transplant, so this will bring a very, very severe consequence. Okay, sometimes it will harm the patients of the, it will harm the life of the patients. Okay, so kidney transplant, yes, definitely is a long-term solution compared to the kidney failure or something like kidney dialysis, because kidney dialysis, right, the patient are required to visit the hospital very frequently. So it's really very inconvenient. And also you need to wait about 3 to 4 hours to just ensure your waste product is being removed to the dialysis machine, right? So this is quite a suffering process for the patient itself. So that's why it's better to have kidney transplant. But the problem is the kidney transplant, right? You need to ensure ensure the kidney transplant are being done in the because very successful. Okay, so you need to match the organ first, match your organ and match the, of this blood type. Okay, so there will be something like the rejection happen is because that the donor itself, it compared to the recipients, they totally don't have the same antigens on the cell surface. So definitely they will always have some immune response. Just like how you're able to reduce the immune response. So that's why if the kidney transplant being done for the patient, right? So the patients, they will need to suppress their own immune response by taking this immunosuppression drug for the rest of their life. So this immunosuppression drug, right, because it's drug, so it causes a side effect. So it will also affect the immune functions of the patient itself. Okay, do you know something like this? Okay, antigen on the cell surface. For your information, okay, every body cell have your own marker. Okay, what I mean by this is something like this. Okay, this is a cell, right? And then this is a nucleus. Okay, then you also remember about on the chapter 3, we learn about the plasma membrane, right? There will have a lot of glycoproteins. Okay, I show you this one. Plasma membrane, there will be a lot of glycoprotein. This one, plasma membrane, the glycoproteins. glycolipid. Can you see this? You can see, right? This is a plasma membrane. And then you can see, right, they have a lot of change because carbohydrate change. So that's why this carbohydrate chain, right, they will become a very unique, requires a identification marker for the cell. So like Ethan and Suyang, if inside your body cell, right, your marker is definitely different in the structure. Okay, they will, I mean, they will not be exactly the same. So that's why it will cause a rejection to happen because of after the kidney is being transplanted to the new patient's the body, right? So the patients, the immune, I mean the immune system, right? They will recognize this kidney is not from our, I mean from their own body. So they will try to reject this kidney because they just think that this kidney is a foreign object to them. So that's why they need to take the immunosuppressant drug. Yeah. Okay, that's all. That's all for this. Okay, so we finished about chapter 13. So let's continue to discuss about some of the answering technique for the paper 4. Okay, so you need to know the waste product. What kind of waste product need to be removed away from the blood by the dialysis is this. They will need to remove away the uric acids, and the ions or salts like sodium chloride, the magnesium, calcium, and any kind of toxins. So usually they will undergo the we call it simple depletion. Pass through the semi-permeable membrane. Okay, this one. Function of the retinal capsules, which is the outer layer in the kidney, is, you can see, right? This is a kidney. Okay, so retinal capsule outer layer is referred to here. Okay, actually it's referred to this whole thing here. Okay, whole thing. Let me draw again. It's referred to this whole thing here. Okay, which include the glomerulus. So it calls retinal capsule. Okay, so it allows the ultrafiltrations to occur. This is because that high blood pressure is generated in the glomerulus because they just receive the blood from the afferent arteriole itself. So this high blood pressure, you need to mention this one, high blood pressure. These are keywords in the glomerulus. So this high blood pressure, it will tend to force, actually forcing the small molecule transport out of the glomerulus and enter to the space of the Bowman capsule. So inside the filter, right, usually it's only the small particle able to get into the Bowman capsule, which include those particles, okay, glucose, amino acid, water, salt, and the urea. So, and also describe the functions labeled as A and B. A is the glomerulus, okay? A is glomerulus, maybe I write here down. Okay, so glomerulus, right, you just need to write about ultrafiltration. This is a keyword for the glomerulus. Glomerulus ultrafiltrations. They just help in remove, I mean, they just help filter away the small particle. And this one, B, they will have this selective reabsorptions. Okay, this B selective reabsorption, they will be the proximal convoluted tubule. Okay, they will have this one, proximal convoluted tubule. So glucose and amino acid, right, they are actually reabsorbed back into the blood. Okay, inside the proximal convoluted tubule. So it's just something like this. Maybe I don't know how, right? You can see, right, this is the regions for the proximal convoluted tubule. Okay, it's at here. So once the blood, right, okay, they pass through from the glomerulus, enter into the Bowman capsule, right? So what are found inside the blood, which is those things, okay, the small particle, which is include the urea, include the mineral salt, okay, glucose, amino acids. Yeah, so they will enter in this proximal covalent tubule, and then they get into here. So what will happen is the selective reabsorption occur. What is meant by selective reabsorption? It's referred to the filtrate, okay? Filtrates inside the tubules, they will diffuse out, get into the blood capillary, like this. Okay, because the tubule, right, is being surrounded by a network of blood capillary, which is for this. You can see, this will be a lot of blood capillary here. The blood capillary is here. That's why they can help to transport. Okay, they can help to transport the nutrients. Okay, reabsorb back to the blood capillary. Okay, so at this proximal covalent tubule, what kind of substance they will be reabsorbed? Which is the glucose and the amino acid. Usually it's these two things, okay? And also glucose is about 100% will be reabsorbed back into your blood capillary. Okay, so they are reabsorbed back into the blood. Okay, so what kind of the excreted substance is removed by the kidney? They contain nitrogen, so it's a urea. Okay, so just remember that urea is the products after protein digestion, because when you want to digest protein, right? So the pathway is just something like this. Proteins is a very, very large particle, right? Large and complex particle. So the proteins, they need to be further break down, become what? Become the polypeptide chain first. After they become the polypeptide chain, right? They will become what? They will become the amino acids. So this is being performed by what kind of enzyme? For the protein to polypeptide chain, right, is being performed by the, we call as the pepsin, right? And then by the trypsins. Yeah, so these amino acids, okay, they need to remove away the nitrogen group, so they will undergo deamination. So after deamination, they will produce ammonia. Then they will become the urea. Okay, so urea is a byproduct of the protein digestion. Urea is also considered toxic, but it's less toxic compared to ammonia, okay? It's still toxic, but it's just lesser toxic as compared to the ammonia, urea. So what kind of structure in the kidney filter the blood? So answer will be the glomerulus. Because C filter, okay, always answer the question based on keyword. Filter is referred to the, we call as ultrafiltration, okay? And also if the component is too large, cannot pass through a filter, it's those components. Okay, so I finished already. For this one, you just have a read by yourself, okay? So all of the answer, important answer, not, and the point is already included in here, okay? So let's try some question, okay? Let's do some question together. Okay, please open this note. Chapter 13, paper 4. The extra exercise for the girls. Okay, after you're ready, let me know. You can let me know.
Ready.
Okay, sure. Let me pick some questions for you to do. So you know how to answer. See, role of the liver in excretion. Just as similar as what we discussed just now. Okay, you need to write about the because deaminations, yeah, which is how your proteins able to remove their nitrogen group away. Okay, this excretory product pass out to the lung requires carbon dioxide. Okay, carbon dioxide. Okay, for this one, we do this question together. Okay. Blood is filter. Name of the part with the letters. Okay, for which one? Blood is filters. Which part? Wait me for a moment. Can you identify which part blood is being filtered? So it will be here. K, okay? Remember about just now we just discussed, right? K, okay? K is we call it cortex. Yeah. Do you know why the cortex functions in the filtration of blood? It's because that the cortex, right, when you look back in the notes, when you look back in the notes, right, let's see the location, okay? Location for the nervous structures. When you see here, it doesn't show in this picture. Okay, sorry, it doesn't show in this picture, but okay, maybe I can show you a actual picture, the global structure in the cortex. It's because at the global, right, they actually locates at the cortex here. Okay, you can see this. You see, this is your whole nervous structure. Okay, and then here will be the cortex around here. Cortex, I put C. Then here will be the vena medulla. Okay, see, here is the vena medulla. I put N here. So you can see the cortex, right? They are located at the upper side of the kidney sections. So the glomerulus, right? They are located at the cortex regions. You can see, right? This is the glomerulus here. Here. They are just located at the cortex regions. That's why it's cortex. And the medulla is is around here, okay? Yeah. This is the cortex, this is the medulla. Then how about when the urine flow to the bladder, right? Okay, this is very simple, it's N, which is the ureter, okay? Which is the ureter. Ureter, and this is N. Oh. And then the blood is carried into the kidney. They are actually referred to the renal artery. Okay? And then one thing you need to know is renal artery, inside they have very high concentration of what? Which is the urea. Okay? They have very high concentration of the urea waste products. Even though they have a lot of oxygen inside. Because the renal artery, right, inside the blood haven't undergo filtrations, haven't undergo this ultrafiltration and the reabsorption. That's why the incoming blood will be high in the urea. Blood flow out is a renal vein, okay? Renal vein. Okay, so substance, see, substance that will be removed away from the blood by dialysis will be those substances, okay, include the urea, include the urea and then the ions, okay, the ions that you can write about, the ion that you need, which is a salt, sodium chloride. You can write about this, sodium chloride. And also the water, okay, you also can write water and the magnesium ion. Okay, this one. Advantage and disadvantage of the kidney transplant compared with dialysis is the advantage, okay? If you're able to get a new kidney, it's very better than you every week you need to visit to the hospital to do the kidney dialysis, right? So that's why this is the advantage. Patients do not need to return to the kidney for dialysis. But the disadvantage will be what? It will be because the rejections happens. Organ rejection, this is low organ rejections. This may cause the rejections of the kidney organs in the patients. Thus, require the immunosuppressant drug. So this will bring the side effect to the health of the patient. Okay, for this one, what's the liquid part of the blood, right? This is very common questions. Okay, you know, right? Our blood, right? 55% is comprised of the blood plasma. Just only 45% is a cellular component. So the liquid part is belong to the plasma or blood plasma. Okay, and then this is excretion. Okay, simple. See, now they want you to describe the function. Okay, for the A, B, yeah. So how you able to describe the functions? So the A, right? Okay, you can describe globus. Okay, so in the glomerulus, ultrafiltration will happen. Ultrafiltration occur. So small molecule with the small particle size, right, will enters into the glomerulus, which is a capillary, and get into the Bowman capsule. Yeah, this is the point. Okay, you need to write that how the molecule able to pass through the glomerulus and enter to the Bowman capsule. Yeah, so this is due to the high blood pressure at the glomerulus, forcing particle push out to the Bowman capsule. Then how about B? Okay, B is proximal convoluted tubule. Okay, so the proximal convoluted tubule, what they do is they will do the selective reabsorption. Okay, so they need to transport the molecule from the tubule back to the blood capillary, such as transport what? Such as the glucose and amino acid. They will reabsorb back. And then for this one, okay, this is a common question we come out for this chapter. They will give you a condition, and then with the glucose, protein, sodium, ions, and urea concentrations. So this is the blood at the X. So at the Y and the urea at the Z. So what will happen is, you can see, right? Name the substance which is the molecule are too large to pass through the wall of capillary. What kind of molecule are too large? Which is the red blood cell, the platelets. Okay, you can talk about protein also. Proteins. So you can see, right, the glucose are like this. So the proteins, when they reach the food at the Y, right, they become totally zero at all. No more protein. So this show what? This show that blood at the X, right, it will be the top glomerulus. So at the Y, right, it will be the requires the proximal convoluted tubule. Okay, PCT. So urine at the Z, right, you can see glucose and the protein disappear already, just left the sodium ion. So this is occur at the loop of Henle. And then you can see, right, the difference between the fluid at Y and urea and Z is, you can see the sodium ions increase, urea increase. Okay, what does this happen? It's because the water reabsorption also takes place in the loop of Henle. So inside the tubule, right, they become lesser water. So if the water become lesser, right, so inside the solid, which is the sodium ions and the urea, they'll be increased in their percentage. So which are all reabsorbed in the kidney? It will be the glucose. Just remember that glucose will be reabsorbed all in the kidney, okay, at the proximal convolutional part. So that's why if your urine, right, if your urine contains glucose, this is totally because this is totally a not good situation, okay. Maybe you have the underlying diabetes, maybe you have the kidney injury, so it cause the kidney, right, the kidney tubule unable to reabsorb 100% of the glucose. It's because that you need to know that 100% glucose, right, will be reabsorbed from where to where? From the tubule into the capillary. And then this process is occur at the proximal convolutional tubule. Which product you refer to this urea? Okay, then they ask you why the concentration of sodium and iron is greater at the Z than Y. So why this urine at the Z, right? They have a higher concentration of the ions and the urea. So the reason is water has already been reabsorbed by osmosis. Okay, because right, you just imagine that inside the urine, right? Maybe I can draw at here. You can just imagine at this place. Reabsorption of water happens at the loop of Henle. Remember this? So if there's too much water being reabsorbed away, it will cause the contents, okay, the contents inside the tubule, right, inside this nephron kidney tubule, become lesser in water, right? So the water will be lesser, so it will increase in their proportions for the salts and the urea, okay? This is because that water has been reabsorbed by osmosis. Remember, water always transport by the osmosis, not simple diffusion, not active transport, only in the osmosis. Osmosis is only involved in the movement of the water molecule. That's all. Okay, so they give you 2 marks, and then 1 more mark is you can write about in the Z. There is no change in the sodium ion, urea, and solid. But volume of water become less. Thus, will increase the concentrations of sodium ions, urea. Okay, here is the answer that you need to know. And then for the execution again, and then, yeah, for this one, filtration of blood again. So I hope you already know that filtration of blood is referred to which one? It's referred to H. Okay, H, which is the nearby the cortex, okay? Because filtration of blood, right, is the glomerulus. Glomerulus, actually, their situation, I mean, their location, they are located at the cortex regions. Right, and then reabsorption of more solid from the filters. This one, a lot of solids on the filter is C, because they involve 100% of the glucose. So it's C, yeah. The reason is they already reabsorbed 100% of the glucose into the blood. And then this one, water is absorbed by the osmosis to determine the concentration of the urine. So this is the G part. Yeah, because water is being reabsorbed from the tubule back to the blood capillary. So this is G. So for the unfiltered blood, right, it will go back to the vein. Okay. D. So this we call as a RENA vein here. RENA vein is at here. This is a RENA vein. Here is a RENA artery. And here will be the afferent arteriole. Okay, so just remember that renal artery, right? They have a very high urea concentration. But the renal vein, right? They have a very low urea concentration. Is because they're already being excreted out in the urine. Okay, so they're low urea, but they still have very high carbon dioxide. Okay, because renal artery and vein, right, inside they contain the blood. Just one is oxygenated. This one, just that one is oxygenated blood, another one is a deoxygenated blood. Yeah, this is a very interesting question. They ask you to list out the component of blood. Okay, is what blood cell found in the filtrate? This filtrate is referred to the urine filtrate. No, I think it's different thing. Filtrate and the urine. Oh, okay, this filtrate is referred to the contents found inside along the tubule, which is inside the proximal convoluted tubule, inside the loop of Henle. Yeah, those inside the content we call as filtrate. After they excrete out the liquid, right, they know as a urine. So inside the filtrate, right, does they contain the white blood cell? The answer is no. Okay, they don't have white blood cell. The white blood cell is too large in size already. Okay, for both also this one is cross. Sorry. Do they have a plasma proteins in the filter, right? Also, they don't have. Urine also they don't have. Because plasma protein, their size is too large already. Okay, glucose inside the filtrate is they have it. But in the urine, they won't have it because they will be 100% reabsorbed by the proximal convoluted tubule. How about urea? Urea in the filtrate, right? Yes, they have it. Okay, in the urine, yeah, they have some of it. Then how about the salt? Salt, right, in the filtrates, yeah, they have this also. So both filtrate and urine, right, they have a same equal amount of the salt. Okay, and the water, yeah. Yeah, and then for this one, dialysis machine, okay, just how we just learned about dialysis machine. So I hope you already know that how dialysis machine works. So they ask you, describe the change occur as a blood flow through the dialysis machine from A to B. This one, A. So this is A, they are connected to your artery, okay? Your artery. And then here is they connect the blood to the veins. So they allow the blood to pass through the vein. So how you're able to describe the structure? So just very simple. So you just mention that our artery is connected to the kidney dialysis machine. So inside they have a lot of semi-permeable membrane. Yeah. So it can allow the substance, okay, which is high molecule to transport to out. Okay. Advantage of kidney transplant is you don't need to go to the hospital to do the kidney transplant. I mean, you don't need to go to the hospital to do the kidney dialysis every day. So this is the advantage, okay? Because you can use a new kidney, just that you need to eat the immunosuppressive drug on it. Okay, so I think I finished for the chapter 13. I hope you are clear, okay? If you don't clear, please refer to this one. 学霸笔记, okay? Please refer to this one, 学霸笔记, because the important notes inside I already put to you. Okay, I already jot down already the important note inside. Yeah, just refer to this one if you feel like you don't understand. Okay. And then I think now is the time we can start about the new chapter, which is the chapter 14. Okay. But I still waiting for the homework for the chapter 13. Okay, chapter 13. Yeah, chapter 11 and 12 you have submitted to me. Thank you. But I'm still waiting for the chapter 13 homework. But it's okay. Take your time first. Take your time first. I understand you are busy also. You have other subject to study also, right? Okay, so let's start the new chapter, coordination and response. So this is very, very long chapter because it's talk about the nervous system, okay? Nervous system. So actually this chapter is about something like this. How your body able to, how the body cell, okay, inside your body, able to connect to other body cell. And then they can work together to perform a reactions, or they can react to the stimuli, and then they produce a response. So this is called a coordination response. Okay, so actually, we talk about the coordination response, right? They are talking about two systems. One is called nervous control, okay, because this one nervous system. Two systems will be discussed in the coordination response. First, it will be what? It will be we call as the nervous system. Secondly, it will be the endocrine system. Then first, we talk about the nervous system first. Okay, we want to talk about nervous system, right? You need to know about how neuron look like. Neuron is a kind of cell, okay? You learned in chapter 2 before. Yeah, see, this is a neuron. Neuron is kind of specialized cell. They only found in the nervous system, inside your brain, and also inside your spinal cord. So this is a neuron, and it's a cell, okay? It's a cell. See, just single unit only is a cell. So actually, neuron, right? When you know about this structure, okay? If the exam asks you to describe about structure, then how you will do it, right? You can say that a neuron cell is long and thin cell, okay? It's very long and thin. Yeah. And then the structure, okay, they contain this cell body. They have this one cell. This one cell is actually a fatty layer sheet. They are surround the essence. So they cause an impulse jump from one gap to another gap in the body sheet. Okay, so this is neurons. So actually we have three types of neuron. What is it called? Sensory neuron. So sensory neuron, what they do is they actually carry the impulse from the sense organs to the central nervous system. Where is the sense organ? The sense organ is they refer to the, for example, skin. Okay, skin, or the eyes, or any kind of sense organ. So they need to collect these informations by the sensory neuron, so they can generate a nerve impulse. So this nerve impulse, right, they will pass through the central nervous system, which is the brain and spinal cord. So inside the brain neuron, right, they are found inside the central nervous system, and they are connect the sensory and the motor neuron. So motor neuron, right, they carry the impulse from the central nervous system to the effector, which is the muscle or the gland. Three kinds of neuron, which is this one, sensory, relay, and the motor neuron. Okay, maybe you can have a look at this. Now, see, you need to know about the structure. You can see the structure. Sensory neuron, right, their cell body is located at the middle way. Okay, so this is called sensory neuron. Okay, it's located at the middle. Relay neuron, right, you can see, right, the cell body, okay, is very, very large. And then they have very, very short axons. So they have a very large cell body, but short axons. This is the feature for the relay neuron. Okay, for the sensory neuron, the cell body will be at the middle. The motor neuron, it just look as similar as you learned before, which is this one, okay? They have a cell body, yeah, those things, sun cell, not very real. So sensory neuron is long. Okay, it's very, very long, super long. So they have a cell body branching up at the middle of the axons. So relay neuron itself is much shorter in their leg. And then they have cell body with many dendrites branching off it. See the cell body, and that's all dendrite. Then the motor neuron, right, they are very, very long, okay? And then they has just a large cell body at one side, along with the long dendrite branching off it. So they will need to connect it to the where? They will need to connect it to the effector. Effector is something like the muscle in the gland, right? Because effector itself, they can produce a response. Okay, so your response, right? We talk about response, we call it as a voluntary and involuntary response. So involuntary response, right, is when you make a conclusion decisions. When you make conclusions, okay, this one, conscious decisions, you will carry out a particular actions before it start with your brain. So this is called voluntary. The involuntary response does not involve in the brains as the coordinator and the reactions, and you do not aware. For example, right, voluntary response is now you are listening to me, okay? You are thinking, you are imagining the process. Involuntary response is you are referring to your inside your body, right? Your intestine, okay? I think just now you just have your dinner, right? So inside your intestine, right, they must be required as a peristalsis, okay? They will do the peristalsis reactions. It's not okay. We call it peristalsis actions, okay? They will do these peristalsis actions, so they will help to move the foods passing along the digestive tracts. So this is called involuntary response. This is your brains did not control it. They happen by themselves. Okay, this reaction is occur by themselves. So they're called involuntary. So both response, right, is very important to ensure our survival. Okay, then now we talk about reflect out. The reflect out and the involuntary response does not involve in the brains as a coordinations reactions. Okay, so this reflect out, right, is something like the in Hua we call it 反射, you know, 反射. Okay, 反射 it will produce this one automatic and the rapid response toward the stimulus. Means that you do not think, you just react on it. Okay. For example, if someone touch your eyes, okay, your eyes will be closed up. Okay. In order to avoid the injury. So your eyes they will just do by themselves. So this is called reflex. Okay, so it does not involve in brains, it just involve in the neuron only. Okay, so they can provide a response which is quicker than any type of nervous response. So this is the characteristic for the reflex action, right? Which is they are automatic, fast, and protective. So this can help to prevent the damage to the body. Okay, by using this reflex action, because you don't have time to think already. You need to act and respond faster. That's why you need to have this reflex. Okay, so this is show a very, very nice picture. It shows about the pathway of the nerve. So let's label, okay? First, you have the receptor on your cell, right? The receptor on the cell usually is located under the skin. Okay? So they can detect the stimulus. Any kind of stimulus, okay? Whether it's pain stimulus or you're feeling cold and very hot. So in this case, there will be the pain stimulus. So the pain stimulus, right? Okay, the pain stimulus, right? They will transport to the receptor cell. So receptor cell, right? They can transport the information, okay, reaching to the brain. Okay, so they will get into spinal cord first. Spinal cord. So you can see, right? This is spinal cord. Okay, this is spinal cord. Here, spinal cord. So reflex, okay, they do not involve in the control of the brain. They only involve in the receptor, neurons, and the spinal cord. That's all. And then this sensory neuron, right, they are connected to the brain neuron. And they connected to the motor neuron and then reach to the effector, which is the muscle. So that's why your muscle, they can do their actions, which is maybe they contract their leg away. Okay, so this is the pathway. Okay, and then you should know how to write the term out, okay? The pain, which is stimulus, is detected by the pain's pressure or touch receptor under the skin. So the sensory neuron itself, they will send the electrical impulse to the spinal cord, which is the coordinator. So the electrical impulse is passed on to the gray neuron in the spinal cord. Gray neuron, right, what they do is they can connect the motor neuron and pass the impulse. Okay, motor neuron, right, they carry the impulse back to the muscle in the leg, which is the effector itself. So the muscle will cause the contractions. Okay, just remember about this pathway. It's very important for you. I put a star here, okay? And then you need to remember that example of effector, it will be the muscle or gland. So they can produce a response. Okay, so that's all for the pathway of the nervous system. Okay, and then now we talk about the synapse. Okay, another interesting topic is synapse. Actually, what's meant by synapse? Just imagine it this way, okay? Inside your brain and your spinal cord, right, there will be millions of the nerve cells. So in order to allow the nerve cells to contact each other, right, they need to be connected. Okay, so they will have this one, synapse. So synapse, right, they are actually referred to the space between this one, pre-synaptic neuron, and the post-synaptic neuron. This space called synapse. Synapse occur between these two neurons. Okay, one is because pre-synaptic neuron, one pre-synaptic neuron, and the post-synaptic neuron. Synapse. Synapse just like the space. Okay, see, synapse is just similar to the synaptic cleft, just an empty space. Okay, inside they don't have any kind of structure, but they only allow the vesicle containing the neurotransmitter get into the receptor on the post-synaptic neuron. Okay, yeah, so this is synapse. So how the impulse is passed across synapse, just like this way. Okay, when the electrical impulse travel along the first axons, this triggers the nerve ending of the pre-synaptic neuron to release a chemical messenger called neurotransmitter to fuse with the pre-synaptic membrane. So with the neurotransmitter, right, they diffuse across the synaptic gap and bind to the receptor molecule, one on the membrane of the second neuron, called a post-synaptic membrane. Yeah. So just imagine like this way, okay. On the pre-synaptic neuron, right, what you have is you have this one vesicle. Okay, you have this one vesicle. So this vesicle, right, they will fuse with the membrane and then they will release all the neurotransmitter in the empty space. Slowly, the neurotransmitter, right, they will bind to the cell receptor here. Okay, they'll bind the receptor proteins at the post-synaptic membrane. So that's why they will trigger response in the post-synaptic neuron due to the neurotransmitter. Not like this. Okay. And then what you need to know is, you should know that this pre-synaptic membrane, right, inside they contain the vesicle, yes. But the post-synaptic membrane, right, inside they contain this one receptor molecule, okay, which is we call it protein receptor. So in order to allow the neurotransmitter bind to this receptor protein, right, the shape also must be complementary. okay, to receptor protein with the complementary shape. Okay, so another thing I want to ask you is you can just imagine that when the neurotransmitter they just pass through the synapse and then they reach the receptor proteins on the postcytid membrane. Okay, so like this just imagine that synapse right, there'll be total synapse is actually an empty space just that there's connection between these two neurons. So there will be a neurotransmitter at here, okay, at here. So what is the process which is the neurotransmitter? Okay, what is the process which the neurotransmitter they can bind to the receptor molecule from the vesicle? I give you three options. Osmosis, active transport, or the simple diffusion. Okay, the question is this one. Okay, how the neurotransmitter pass through whole pre synaptic neuron to post synaptic neuron. So at the they want to have a guess how the neurotransmitter they can pass through this synapse and get to the receptor proteins. Is active transport or osmosis or the simple diffusion? Which one? This is asked in the question before, okay? During the exam, yeah, asked in the exam before. Which one? Simple diffusion?
Diffusion.
Or, yeah, that's it, simple diffusion, okay? It's because why? It's because that the pre-synaptic neuron, right, inside they have very high concentration of the neurotransmitter as compared to synapse. Totally very low concentration because they don't have any neurotransmitter. So just as simple as that. It's happened by the simple diffusion. That's all. Okay. So whenever the neurotransmitter, right, they diffuse across the synapse and they bind the receptor proteins on the postcytid membrane, right? So due to the stimulus the second neuron generate. Okay, so they will produce a new nerve impulse at the postcytid neuron, travel down to the second axon. So after the neurotransmitter being used up, can they go back to the vesicle? Can they go back to this pre-synaptic neuron and package back into the vesicle? The answer is no. Okay, no. We can allow neurotransmitter, right, just flow in direction like this, forward only, like this. Okay, which is direction is from the pre-synaptic neuron to the post-synaptic neuron. Cannot be post-synaptic neuron back to the pre-synaptic neuron. Cannot. This is impossible. Okay, so it's one-way direction flow, just like the nerve impulse. Okay, the nerve impulse inside your neuron cell, when they transmit the nerve impulse, they only in the one-way direction only. Same to the blood flow. Okay, blood flow we only want to transfer in one direction. We want to prevent any backflow happen. That's why we need to have the valve. So, how your body able to remove the neurotransmitter away since they don't have able to go back to the pre-synaptic neuron, so they will be destroyed. Okay, they will be degraded by the enzyme sometimes because they want to prevent this neurotransmitter stay too long on this synapse and also bind to the receptor protein too long already. Okay, so this is not good. This is not good. So you want to prevent the continuous stimulation of second neuron. So the present in synapse, right? I mean, the purpose of the synapse is they just want to make sure the impulse only travel in one direction. Okay, just forward, cannot be backwards. Another thing I want you to know is You know, example neurotransmitter. Okay, example neurotransmitter, right, which is quite common they talk about nowadays is dopamines. Have you heard about dopamine before? This one. Have you heard about this before? It's a happy hormones. Okay, it's a happy form of the neurotransmitter because dopamine, right, it will give you a kind of like a positive emotion, dopamine. So this dopamine is a kind of neurotransmitter. Okay, so actually this neurotransmitter, right, they can affect your mental. Okay, they can affect your mentality. So neurotransmitter itself is actually just something like the substrate, okay? Because they have their specific structure. So they only can bind to the receptor proteins with the complementary structure. Okay, so you need to know how to describe the structure. So you can see, right? Yeah. When the impulse arrive at the presiding neuron, see this one. When they arrive at the presiding neuron, this will cause the vesicle to move forwards. And then they will fuse with the presynaptic membrane. And then they release the neurotransmitter to the synaptic cleft, which is a space between the neuron. The neurotransmitter, right, they diffuse across the synaptic cleft down the gradient. Okay, yeah, down the gradient because it's simple diffusion, right? It's from the high to the low concentration. That's why it's down the gradient. So the neurotransmitter, they just attach to the receptor proteins located on the surface of the postsynaptic membrane. So this will trigger the formation of the new impulse, which is travel along the postsynaptic neuron. Yeah, so on the postsynaptic neuron, congratulations, they can finally generate a new impulse. But at the end, this neurotransmitter, right, they need to be clear of. They cannot stay in the synapse too long. Okay, it will bring a very, very bad effect if they stay in the synapse too long. You become addicted, you know. So I think you heard about the drug before, right? Like the drug, like the what kind of drug, right? Like the heroines, okay, morphines, the drugs like this, 毒品 of course 毒品, okay. So actually this drug, right, they're actually playing your neurotransmitter. Oh, this is cocaine, those drugs. They're actually playing to the neurotransmitter. So they will bring a very, because a euphoria effect to your body, okay. Neurotransmitter. Yeah, this one. You will not learn in your IGCSE syllabus, but you will learn in your A level. I'm not sure you study A level biology. I'm not sure you will study this or not in your future time, but it's something like this. So you can see, right? This is the presynaptic neuron, and this is postsynaptic neuron. So you can see, right? Presynaptic neuron, they have a synaptic vesicle. Inside, they call it a neurotransmitter. Just imagine that inside of the this one, inside of this vesicle, right? They contain a neurotransmitter, which is the dopamine. Okay, actually, dopamine is a kind of the neurotransmitter. Dopamine, I think you heard before, right? 多巴胺. Yeah. So this dopamine, right, in the normal situation is they want to transport these dopamines, okay, from this synaptic vesicle and reach to the whole synaptic neuron on the dopamine receptor. Okay, but if someone they have this cocaine, okay, means like if they take the cocaine drug, what will happen is the cocaine they will compete. Okay, they will compete with the active site. It's not active site. They will compete with the binding site. Cocaine they will compete to the binding site on the receptor proteins. So it will prevent the dopamine from binding to the receptor proteins on the dopamine receptor. Okay, so this will bring a big, this will bring a very, very bad effect. Okay, if you prevent the dopamine from binding to their receptor. So neurotransmitter cannot be prolonged in the synapse. They need to be recycled back or destroyed. Okay, that's all. And then we talk about the sense organs. Okay. Before I talk about this, right, let me show you about chapter 14. Okay. What are the important notes included in the chapter 14? This is okay. Data I sent to you. Chapter 14. Okay. We talk about the sense organ first. Example of sense organ is they can detect the change in the environment and stimulate the electrical impulse in the response. Means that this sense organ, they can sense any change happens in the surrounding environment. Okay, they can collect the stimulus and then they bring back to your brain or spinal cord. Okay, so you can produce a electrical impulse, and then okay, from the sensory neuron to the relay neurons, which is inside your spinal cord, and then they reach to your motor neurons and then to your effector. So effector, they can carry out the response. So what other kind of sense organ include the skin, tongues, nose, ear, and eyes? But we just learn about these two types only. We learn about skins and the eyes, that's all. We don't know about tongue, nose, ear. Okay, but you should know about function also. Skin is very sensitive to pressure, heat, and the cold. It's because that there's too much receptor located on skin, include what? Pain receptor, thermoreceptor, okay, mechanoreceptor. Those receptors are located on the skin. For example, like this, pain receptor, thermoreceptor, mechanoreceptor, which is the pressure. They can help to detect the change in the pressure. For the eyes, right, it's sensitive to the lights. Okay, so that's all. Okay, for this structure of eyes, right, okay, it looks a bit complicated for you, okay, but for this part, right, IGC exam, they're very, very rare to come out this eyes part, but you should know also, okay, you see, this eyes, right, you should know about the structure, okay, structure of the eyes itself. So actually, the eyes, they comprise of 3 layers, which is from the outermost layer, which is the sclera, and then the retina. And then on the front part, right, there'll be as this one, pupil, and then this lens. Okay, lens is connected by the ciliary muscle. Okay, lens will be connected to the ciliary muscle by the subspecial ligament. Okay, what's ligament? Ligament is a very strong fibrous tissue. It's very, very hard to break it. Ligament. Okay, just similar like the tendon. Okay, okay, this one you learn more in future. Okay, ligament just something like this. Let me show you ligament. You have a ligament on the ankle also, like this. See, ligament. Okay, you can see right. This is a ligament. It's a type of connective tissue. Usually, they help to connect from bone to bone. So in the eyes, right, you also have a ligament. Just they help to connect from the muscle to the lens. So the cornea itself, okay, cornea we call it as 瞳孔. Okay. It's a transparent lens, right? They can reflect the light. Okay, cornea just look like the mirror because they can help in the light refractions. For the iris, they can dilate or they can constrict. Okay, so they can control how much light enter the pupil. So for the lens, right, they can change their shape. Means that they can appear longer and thinner or shorter and thicker. Change. They can change the thickness of the lens, either become longer or shorter, thicker. In the retina, okay, they contain the light receptor cell, which is the rods. So these rods, right, they can detect the light intensity and the cones, which is detect the color. Okay, this one you can put a star here. This is the favorite exam questions. Okay, this is a common exam question. Retina is at here. Where is the retina? This is retina, okay? Retina is actually located at the, we call as the innermost layer. They are facing at here. Can see, right? This is retina. Here. Okay, sorry. Here. This is retina structure. Okay, inside the retina, they have a lot of light receptor cell. There's two light receptor cell, okay, rods and cones. Rods, they can help to detect the highlight intensity in the daytime. Oh, sorry. Rods, they can help to detect the highlight intensity in the nighttime. Not daytime, it's nighttime. You can just imagine that at the nighttime, right, you want to go toilets. And then there will, at your house there will be a lot of light already shut off, right? I mean, there'll be a lot of darkness, okay? So you still can see clearly the surrounding object. So why you can see clearly is because the rods help you to detect the light intensity. They can help to collect the light in the nighttime. Means that there's no presence of the lights. Okay, how about the cones? Cones, they can help to detect the color in the daytime. In the daytime. So rod is for the nighttime because rod is very sensitive towards the highlight intensity. For the cones, it will be the daytime because they can detect the color, which more variety. Okay, the optic nerve is referred to the sensory neuron. Sensory neuron is connected to here. This one here. Here is the optic nerve. Okay. Optic nerve, right, is a nerve connected to the eyes. So they can carry the impulse between the eyes and the brains. For the pupils, okay, it's a hole. They can allow the light to enter the eyes. Okay, so this is a pupil. So just need to know about the important part will be this one, retina. Okay, they connect the rods and the cones, and then optic nerve. And then will come on retina, optic nerve, and also here, ciliary muscle and the suspensory ligament. Usually they'll count these four structures. Okay, fovea also. Okay, fovea also. This five structure. Okay, you can see, right? Later, let me show you another thing. Okay, it will be. I think I open this note to show you better. Okay, this one. Okay, this one I'll send. I will send to you later. Okay, I will send to you. You can have a look. Okay, this note already summarized all the important point for the chapter 14. Yeah, you can see, right, for this one. Let me show you. Okay, so this rod and cones, right, they are just located at the retina. So they contain this photoreceptor. Okay, so rod is very light sensitive, especially at the night time. Okay, and also for the fovea. Okay, in the fovea, right, fovea is actually located. Actually, it's a spot. It's a spot located on the surface of retina. But in the fovea, right, they only contain the cones. They only have these cones able to detect the color light. So this is in the fovea. Okay, so the exam will ask you about the rods and cones. So what kind of answer you should write is you should write that rods and cones, right, both are also photoreceptors and located in the retinas. So they can detect the light. So the rods, right, okay, they help to detect the lights, especially at the night time. So it's much more sensitive. Actually, the rods, right, rods is a photoreceptor, which is much more sensitive as compared to the cones. Okay. So rods is for the low light visions, which is very sensitive at the night time. So how about the cones? Cones will be the color visions and it's very high resolution details in the bright light. So cones, right, they can help to differentiate the colors. So this is not found in the rods. Okay, rods, the function is definitely much more sensitive to the lights than the cones are, because at the nighttime, right? In the nighttime, I mean, at the dark area, right? You need to collect all those we call it information, right? So that's why rods is very sensitive compared to the cones. So they can allow us to see in a very dim conditions, which is very dark condition. You still can detect where you are. And then you also have the blind spot, okay? Blind spot usually is located very near to the optic nerve. So at this blind spot, right, there will be very lack of the light sensitive sound, okay, resulting in the area where there's no image being perceived. Let me show you the area of blind spot now, later. Blind spot. You should know about this also, blind spot. Blind spot on the eyes, which is at the retinal area. See, here. Here will be the blind spot. Here, okay? See. Which is the location that optic nerve exists. Okay, at the blind spot, right, you totally don't have the rods and the cones. That's why you cannot detect anything. There will be no any kind of the photoreceptor. Okay, I put here, yeah, the blind spot. Okay, so I think we could continue in the next class for this chapter 14. So please complete your chapter 13 slot. Chapter 13 slots. It's not notes. Okay, your exercise, paper 2 exercise. Okay, complete your chapter 13 paper 2 exercise, and also the chapter 11. Okay, so I will need to mark the chapter 11 and chapter 12, and then I'll get back to you. We can discuss in next class, and also the chapter 13. Okay, so that's all. That's all for the class today. See you in next class. So do you have any question to ask me now? Okay, everything good? Clear? Understand? Okay, okay. Sure, sure. Bye, bye. See you next class. Bye.